we were working with each other in the last session of the lecture part, and today we will discuss with each other on the linkage disequilibrium, which is the nightmare of all the geneticians. We'll discuss with each other also on the population genetics, on a little bit introduction on epigenetic, which is a very interesting field. And yeah, I know it's 1 p.m. So okay. And in the last part, we will discuss with each other on the epigenetic, which is a really interesting part to decide how on the mechanism how the body can decide which genes was turned on and which gene was turned off in a different type of cell type and in a different period of time. And if we still have time, we will have a quick recap. I will introduce with you very briefly what we should remember in all the 7 chapters, in all the 10 sessions that we have discussed from the beginning. But first of all, we will come to a small introduction on the CRISPR-Cas9, a very powerful tool to edit the genomes. This research was launched by 2 scientists, Jennifer Doudna and Charpentier, and their research have won the Nobel Prize, as I remember, in 2020. And this tool is developed from a mechanism of a bacteria to defend against the attack of a virus. So let's see how. Wait a minute, so I can share cell. Yeah, cells.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called Rna, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognized that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories, or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things, like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented litter mates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way that DNA is repaired after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known. But it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos, to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s, when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So Genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Jennifer, this is a technology with huge consequences. You pointed out your attitude about asking, you can call it a pause or a moratorium or a quarantine, is incredibly responsible. There are, of course, the therapeutic results of this, but then there are the non-therapeutic ones, and they seem to be the ones gaining traction, particularly in the media. This is one of the latest issues of The Economist. Editing humanity is all about genetic enhancement, it's not about therapeutics, right? What kind of reactions did you get back in March from your colleagues in the science world when you asked or when you suggested that we should actually pause this for a moment and think about it?
My colleagues were actually, I think, delighted to have the opportunity to discuss this openly. It's interesting that as I talk to people, my scientific colleagues as well as others, there's a wide variety of viewpoints about this. So clearly it's a topic that needs careful consideration and discussion.
So there's a big meeting happening in December that
Correct.
You and your colleagues are calling together with the Academy of Science and others. What is your expectation? What do you hope that will come out of the meeting practically?
Well, I hope that we can air the views of many different individuals and stakeholders who want to think about how to use this technology responsibly. It may not be possible to come up with a consensus point of view, but I think we should at least understand what all the issues are as we go forward.
Now, colleagues of yours like George Church, for example, at Harvard, they say, yeah, ethical issues basically are just a question of safety. We test and test and test again in animals and in labs, and then once we feel it's safe enough, we move on to humans. And so that's kind of the other school of thought, is we should actually use this opportunity and really go for it. Is there a possible split happening in the science community about this? I mean, are we going to see some people holding back because they have ethical concerns and some others just going for it because some countries under-regulate or don't regulate at all?
Well, I think with any new technology, especially something like this, there are going to be a variety of viewpoints. And I think that's perfectly understandable. I think that in the end, this technology will be used for human genome engineering. But I think that to do that without careful consideration and discussion of the risks and the potential complications would not be responsible.
Now, there are a lot of technologies in other fields of science that are developing exponentially, pretty much like yours. I'm thinking about artificial intelligence, about autonomous robots, and so no one seems, aside from autonomous warfare robots, nobody seems to have launched a similar discussion in those fields and calling for a moratorium. Do you think that your discussion may serve as a blueprint for other fields?
Well, I think it's hard for scientists to get out of the laboratory, speaking for myself. It's a little bit uncomfortable to do that. But I do think that being involved in the genesis of this really puts me and my colleagues in a position of responsibility. And I would say that I certainly hope that other technologies will be considered in the same way, just as we would want to consider something that could have implications in other fields besides biology.
Jennifer, thanks for coming to TED. Thank you very much.
Thank you.
So the CRISPR tool, the original of the CRISPR tool, it comes from bacteria. Actually, it's a defense system of bacteria against the infection of a virus. So when a virus injects its inherited material, it could be the RNA, it could be DNA, into a bacteria will using its sequences to make some kind like a cell memorial. And the next time the virus inject its inherited material into the bacteria, it will use CRISPR to cleavage the genetic material of the virus. based on its memorial, based on the sequence of memorial. And scientists have applied this tool to cleavage the fragment of DNA of interest. And like we have studied in chapter 4, in cell, we have the DNA repair mechanism. So whenever a fragment of DNA was destroyed, it will repair the DNA as much as possible. And when the scientists inject artificially synthesized DNA fragment, it will fix in the region, so they can successfully replace the DNA, the old DNA, by the new one that they design. And up to now, there are applications of genome editing by CRISPR in plant model, in plant cells, in bacteria, in human cells. But however, the genetic genome editing in the human embryo tissues were not allowed yet. It's not due to the technical problem, but more than the ethical problem. So in here, in your stage, we have a research team also perform research on CRISPR to design again the genomes of plant species in rice. So just some information I want to provide you. All right, so let's come to the next chapter. Okay, so in this chapter, we will discuss with each other on the linkage disequilibrium and the difficulty it brings to genetics. And also linkage disequilibrium was provide also a powerful tool to identify the genetic region that related to one disease. So first, let's come to linkage disequilibrium. So you have all studied around linkage disequilibrium in high school. So I will just have a very brief recap. So in the time of Mendel, in the last chapter, we have studied that he have invention his 3 rules of inheritance. One among those was the law of dominance, the law of separate inheritance, and so we know that in the 7th trait here, he had chosen the 7th trait that followed the 3-to-1 ratio, follow his rule. However, after nearly 100 years, another genetician, Thomas Hans Morgan, who the first US citizen to win the Nobel Prize, who also considered as the father of modern genetics, when he chose to learn his study in the fruit fly, or in scientific name Drosophila melanogaster. So this species was chosen for his study based on, first, it have a really simple organized of its genome. In fruit fly, there is only 4 pairs of chromosomes, 3 autosomal chromosomes and 1 pair of gender chromosomes. And the life cycle of fruit fly is quite short, so the scientist can easily to observe the consequences of any cross, because the life cycle of fruit fly is just within around 2 weeks. So even nowadays, many labs are still using fruit fly for research for such reasons. So Thomas Morgan had performed, he has chosen the same experiment like the experiment of Mendel, when he has crossed the two breeding fruit fly, one with a yellow body and the light long wings here, and crossed with another fruit fly with a black body with short wings. And this two fruit fly is true breeding, so he's sure that is they are homozygous of the concerning allele. So in F1, we obtain 100% was carry the dominant allele, the dominant phenotype, as well as the law of dominance of Mendel. So there is no surprise here. But when Thomas Morgan bring a back cross, that means he take the F1 here and cross back with one of the parent, which carry the recessive phenotype, and he recognized that in the population BC, that the offspring, the proportion of the population BC is different with the theory of Mendel, in which the parental phenotype it takes around 80%, divide equally to the two phenotype, one with the both dominant phenotype is 40%, and the other with both the recessive phenotype is 40%, is black body and the short wing. And there's two new phenotype, which is recombinant phenotype, which is black body with the long wings, and yellow body with the short wing. And the percentage of these two phenotype is 20%, and divide equally by 10% of the black body with the long wings, and another is 10% with the yellow body and the short wings. So at the time, if the population BC is following Mendel rule, Mendel third rule, the rule of independent assortment, we can see that if it follows this rule, the percentage of each gamete will be 25%. So this is the F1. This is the gamete of the F1. And this is the recessive parent. So the percentage of gamete will be 25% of big B, big S, and for the yellow body with the long wings, 25% with small b, small s, which is the black body with short wings, 25% small b, big S, which is black body with the long wings, and 25 other percent is yellow body with short wing. So the ratio should be 1 to 1 to 1 to 1. It follows the third rule of Mendel. But in fact, the result was bring down with the 80% of the non-recombinant phenotype, which is carried to phenotype of parent, and 20% of the recombinant phenotype. So actually, at the moment, Thomas Morgan don't know what is going on, and he had not successfully provide a suitable mechanism to explain this until a dozen years later, one of his students, Mr. Alfred Stoverman, one of the Morgan students, when he did homework, he's figured out everything. So the mechanism that Stoverman provide is in here, the two PEP allele corresponding to the color of the body and the length of the wings is located in the same PEP chromosomes. And during DNA replication, somehow in for a certain proportion, there is a cross-link between this pair, and the recombinant rate in this case is around 20%. So it 20%, there's the proportion of the change to happen a cross-link between these two positions, and the result is the gamete will divide by 20% for the two recombinant gamete, and 80% with the non-recombinant gamete. And in the parent, the gamete is more or less the same, because even there is a cross-link happen here, there is no change in the outcome, because this is homozygous of the allele. So even their change, there's nothing changed in the gamete. So as a consequences, when the gamete in here combined with the gamete of the parent of the, we will have a population BC is 80% of the non-recombinant and 20% of the recombinant, and divide by each pair allele. So he have named this unit is Morgan, because he want to show his respect to his supervisor, to his teacher. So he called that the genetic distance between these two genes is 20 centimorgan. And Morgan is the name of his teacher, which is corresponding to 20% of the ratio of recombinant happening between these two locus. Okay, so dear my student, in the future, if you have invented anything, you're very welcome to name it after me. Thank you. Anyway, so for example, in here, if we call that the distance, genetic distance between the two positions B and S is 27 centimorgan, that means that the percentage of cross-link in this between these two positions is 20%. And if we have the genetic distance between pair allele is 30 centimorgan, that means there is 30% of crossing was happened between this position, two positions in meiosis. Okay. So just a quick reminder about the meiosis. So in meiosis, the cell will become one time the DNA was replicated, and two times the cells was divided. And as a consequences, their genomes will be decreased by half in the daughter cells. And in meiosis 1, during the metaphase, the two the pair of double chromosomes will gather around in the metaphase aqueous, and that is the time where the cross-link over was happening, when a part of the homologous regions in this chromosome will exchange to its sister chromosomes. And normally, the genetic distance will have a correlation with the physical distance. So genetic distance is the percentage of exchange, and the physical distance is the base pair. The physical distance is calculated at the angstrom, and angstrom is equal to the pair corresponding to the pair of chromosomes between the two locus. So theoretically, physical distance is correlated with genetic distance, but it's not always true. Since the maximum frequency of crossing over, the maximum frequency genetic distance is about 50 centimogon. And in case of 50 centimogon, we come back to the rule manner, is independent assortment. So here is a small exercise for you to remind you what is the genetic distance between the two allele. So I have the backgrounds here. I have the genotype here called the B is corresponding for the color, and the S is for the length of the wings. So in here, please calculate for me the proportion of the population BC if the genetic distance between the B and S allele is 30 centimogon, and in the other question is 0 centimogon. That means is what is in Thai linkage. Shall we? We all have studied this in high school. So any answer for the first question? What is the proportion of the population BC if genetic distance between this is 30 centimogon? Guys, are you all fall asleep? Anyone? You can type the answer in the chat box for me. All right. Okay, so maybe you have forgot all about the high school, or you just too lazy to answer me. All right. So if the first question is if the genetic distance is 30 centimogon, so we will have the distance in here, the percentage of recombinant gamete in here is 30%. And the other is 70%, there's no crosslink, right? And because the percentage is distributed equally between the two paired chromosomes, so we will have 50% of this recombinant, that means the black body with the long wings, and 15% will have the yellow body with the short wings. And in the other case, with the parent phenotype, we will have 35%. We'll have a yellow body with long wings, and the other 35% will have the black body with the short wings. Okay. And in the other case, sorry. In the other case, when we have the genetic distance between those is 0 centimogon, so we will have 0% we'll have a recombinant phenotype, and we have in here the parental phenotype E100% distributed equally between the two. So we will have 50% yellow body with long wing and 50% of black body with the short wings. All right. So next is the gene mapping. How we can identify the gene mapping, that means the genetic distance between the PrEP genes, based on the ratio of recombination or the percentage of cross-link order. So in here, I have 3 pair of genes, A, B, and C. And I have the genetic distance between A and B is 25 centimorgan based on the test, the cross-link test. I have also the genetic distance between the AC and the BC is 16% and 19% consequently. So from this, please identify for me if we have the order of gene is ABC, that means the B is located between in the middle of A and C, or if the A is located between B and C, or the C is located between A and B. So to handle this, first we come from the largest number. In here is 25%. So I will draw an A and B on the genome with the genetic distance is 25%. Okay. So if we have this, I can have the next is the genetic distance between A and C is 16 centimorgan. So we will have C can be over here, which is 16 Morgan distance between A, or located in here with the genetic distance is 16 Morgan. But again, I use the light data, which is the genetic distance between B and C in 19 centimorgan. So because it's 19 centimorgan, so I have C must be here. Because if C is here, the genetic distance between B and C is much more than 25%. So the right answer is the rule is A and C and B. And one notice that we have no additive rule in the gene mapping. So for example, here I have this genetic distance between A and C, which is 16 centimorgan, and BC in 19 centimorgan, and we cannot take the genetic distance between A and B is equal to the sum of genetic distance AC and BC, because we can see here it's only 25. It's different with 16 plus 19. Okay. Again, please, if you send me a question, please send it to everyone. Okay. And just like I just explained to you, there is no additive rule in gene mapping. Okay. There is additive rule in physical distance, but there's no additive rule in genetic distance. So in here, the genetic A and B is different with the genetic distance between A and C and B and C. Okay, so next exercise, please draw a map here for the 14 for me. Know that the recombinant frequency, the ratio between A, B, C, D is like provide in the table, please. By the same rule. Anyone finish? Anyone can provide me the answer for this? So in here, BADC, CDAB. Actually, CDAB is the same with BADC. So we doesn't have the exact sort of the B come first or the C have to come first. All right. So with the same rule, we have BC is the longest distance. So I will draw B and C's. B and C, we illustrate one with 25 centimorgan, right? So the next data is the distance between A and B is 25%, and distance between B and D is 30%. So A in this is located between in the middle of BC, because in here BC is the longest. But if the BAD or if the BDA, so in here the distance is AB is less than the distance between BD. So A is close with B is more than D, right? So we have the distance between AB is 25 centimorgan, 20, sorry. And the genetic distance between B and D is 30. So we have the distance AD is 15, the other in here is 25, is 20, all right? Right. So, okay, I will get away of my So the right answer is B, A, D, and C. So again, with the genetic distance, so this is the first genetic distance that have been provided in the fruit fly. With the first phenotype is the length of the iris state, a little antenna in the heart of the fruit fly. So the next is the phenotype related to the color of the eye is around 16.5 centimorgan ahead. Okay, so when they see that, we can see that the percentage of recombinant between these two phenotypes is 16.5%. But again, when we draw a map like this, we will have the genetic distance in here is 48.5, and the genetic distance here is 57.5, 67 centimorgan. So what does that mean? We just discussed a few slides later that the genetic distance maximum is 50%, dealing to 50 centimorgan. But in here, we have several phenotypes have the genetic distance more than 50 centimorgan. So what does that mean? So what is the meaning here? Okay, so when they identify the order of the phenotype, they will continue to calculate, for example, the genetic distance between the maroon eyes and the black body in here is, can you calculate for me? The genetic distance in here is 35 centimorgan. And because the genetic distance...
excuse me, I thought that it is not additive. It is not additive.
Yeah, it is not additive. It's not additive. It just yeah.
But why is it 32? Like it cannot be a different value.
Okay, I will explain what is not additive. So, for example, in this tree, now let's focus on this tree. Okay, just for simple. Okay. So in here we have the percentage of cross-link between this pair is 16.5%. Okay, you understand this part. And we have the distance between this pair is 32. So that means what? That means that in here I have the genetic distance between here is 16.5 centimorgan, and the genetic distance between this continuous with these two continuous pair is 32 centimorgan. But so I take this here is a beginning, like a point of compare, and we take this is 48.5 centimorgan. It's not corresponding that the percentage of exchange between this pair, if I perform a cross between this pair, it will not 48.5%. Okay, it just corresponding to the genetic distance. You understand this point? It quite important. It's the same question with why in here we have the genetic distance with 57.5. Theoretically, we never have a genetic distance more than 50% of centimorgan, in which 50% of percentage of cross-link is the maximum percentage of cross-link due to the independent assortment parameter. But we have it in here because we have a percentage proportion between these two is 9%. That's all. You all understand this part, or? So you want me to re-explain this in Vietnamese, maybe?
Yes, please.
Okay. Right. Wait for me a minute. Ok. Rồi ở đây khi nói về liên kết gen ý, thì mọi người đều biết là cái tần số hoán vị gen lớn nhất là 50%, và 50% ứng với là phân ly độc lập tử hợp tự do như là Mendel, đúng không? Nghĩa là hai cái cặp gen này không hề liên kết gì với nhau cả. Về mặt lý thuyết là không bao giờ có tỷ lệ hoán vị gen lớn hơn 50%, đúng không? Cho nên là khoảng cách di truyền giữa hai cặp alen lớn nhất là bằng 50 cm/organ. Ok. Ok. Tuy nhiên khi mà đề cập đến cái lĩnh vực gọi là gene mapping, có nghĩa là bản đồ di truyền ấy, thì người ta sẽ bắt đầu mark những cái cặp tính trạng. Ví dụ ở đây tôi có cặp tính trạng đầu tiên, tôi quy ước nó là 0, tôi lấy nó là mốc 0, thì cặp tính trạng tiếp theo gần với nó nhất là 16,5 cm/organ. Điều đấy có nghĩa là gì? Có nghĩa là tần số trao đổi chéo ở đây là 16,5 cm/organ, 16,5%. Ok. Tiếp theo, cặp tính trạng tiếp theo là tôi có ở đây là cặp tính trạng giữa màu mắt và màu thân này, là khoảng cách di truyền là 32%, có nghĩa 32% xảy ra trao đổi chéo ở đây, đúng không? Thì tôi biết là khoảng cách di truyền giữa cái cặp liên tiếp này là 32 cm/organ. Và vì tôi đã quy ước cái tính trạng đầu tiên là 0 ở mốc 0, tính trạng thứ hai ở mốc là 16,5, cho nên cặp tính trạng thứ ba tôi sẽ quy ước nó là 16,5 cộng thêm với 32, tức là 48,5. Điều đấy không có nghĩa là cái tần số trao đổi chéo giữa cái cặp thứ ba này với cặp thứ nhất là 48,5%. Mà đây chỉ là cái cách để tôi xác định cái khoảng cách di truyền. Tôi vẽ dựa vào đó để xác định cái khoảng cách di truyền. Có nghĩa là cái bản đồ di truyền này chỉ có ý nghĩa để xác định tần số trao đổi chéo khi chúng ta tính giữa hai cặp tính trạng liên tiếp. Ví dụ tôi muốn tính là khoảng cách di truyền giữa cặp này với cái cặp tiếp theo nó là cặp này là bao nhiêu, thì tôi lấy 75,5 trừ đi 67, có nghĩa là ra là bao nhiêu?
Teacher, I had to interrupt you, but where's your screen?
I have not
Can you share your screen again?
I have... All right, I forget it. All right, sorry. Okay, again. Rồi, ở đây tôi có bản đồ gen, bản đồ di truyền giữa một loạt các cặp tính trạng, đúng không? Thì tôi lấy cặp tính trạng đầu tiên là cặp tính trạng về độ dài của cái râu ở trên đầu này. Tôi đặt nó ở mốc 0. Được chưa? Cặp tiếp theo gần với nó nhất là cặp tính trạng màu mắt. Thì sau một phép like thì tôi tính ra tần số trao đổi chéo là 16,5%. Vậy tôi vẽ lên một cái bản đồ di truyền và tôi thấy cái cặp này, tôi quy ước nó là cái cặp này là 16,5 cm/gam. Được không ạ? Ok. Tiếp theo tôi tiến hành tiếp một phép like nữa thì tôi thấy là cái cặp tính trạng tiếp theo mà gần với cặp màu mắt này nhất là cặp tính trạng là màu thân. Và cặp tính trạng màu thân này tỷ lệ xảy ra trao đổi chéo là bao nhiêu? 32%. 32% xảy ra trao đổi chéo. Có nghĩa là khoảng cách di truyền giữa hai cặp này là 32 cm/gam. Mà cặp này tôi đã quy ước là 0 này. Cặp này tôi quy ước là 16,5. Thì cặp này tôi sẽ quy ước nó ở vị trí là 16,5 cộng 32, tức là 48,5. Được không ạ? Nhưng điều đấy không có nghĩa là khoảng cách di truyền giữa cái màu thân này với cả cái cặp mà tôi quy ước là ở mốc 0 đây là 48,5%. Tần số trao đổi chéo ở đây không phải là 48,5%. Nó sẽ là một con số nhỏ hơn. Đúng không ạ? Mà tôi chỉ ghi 48,5 ở đây là khoảng cách khi mà tôi... Cái điều này nó chỉ có ý nghĩa khi mà tôi so sánh hai cặp tính trạng liên tiếp. Ví dụ ở đây tôi so sánh cặp tính trạng đây là độ dài cánh với cả cặp tính trạng ngay gần với nó là hình dạng của cánh. Thì tôi thấy ở đây là tần số trao đổi chéo giữa nó với cả cái giữa hai cặp này là 75,5 trừ 67 là bằng 8,5%. 8,5% xảy ra trao đổi chéo giữa cặp này. Điều đó không có nghĩa là cặp này cái tính trạng này tần số trao đổi chéo với cả cái tính trạng ban đầu đây là 67%, không có. Tần số trao đổi chéo lớn nhất chỉ là 50%. Cái này là rất rõ rồi, đúng không ạ? Ok. So,
mà một chút có nghĩa là mình chỉ phải quy đổi giữa cái tính mol và phần trăm với những gen đi liền kề nhau, đúng không?
đúng rồi, chỉ có thể quy đổi như thế với các cặp gen liền kề nhau.
Thế nếu như mà đặt ở trên một cái đường như thế này thì nó sẽ được biểu diễn như này, nhưng nếu mà đưa vào bảng thì nó vẫn sẽ theo cái quy luật là dưới 50, đúng không thầy?
Đưa vào bảng là theo quy luật dưới 50 là sao em?
Là cái table giống như cái bài lần trước, cái bài ngay trước đấy của thầy thì nó sẽ không bao giờ hiện giờ.
Ok, thế thì nó sẽ xuất hiện một cái chuyện là như thế này. Ok, chờ thầy tí, thầy sẽ dở một cái file pen ra. Ok, thầy xin phép được tắt tiếng em. Ok, khi mà người ta ở chương 4 thì chúng ta học thì chúng ta đã biết về cái gọi là genetic marker rồi, đúng không ạ? Thế thì người ta sẽ tính genetic marker dọc hệ gen người, ví dụ như là dọc hệ gen người. Ở đây tôi có hai cái marker 1 và marker 2, ví dụ như vậy để cho nó dễ. Và sau khi một phép like thì tôi tính toán được khoảng cách di truyền giữa hai marker này là bao nhiêu? Là ví dụ 20 cm, đúng không? Nếu như tôi chỉ biết đến hai cái marker này thì tôi sẽ nói là bộ gen người có khoảng cách là 20 cm, vì ở đây tôi chỉ thấy hai marker thôi. Tuy nhiên sau đó thì qua các dự án giải trình tự hệ gen người, tôi phát hiện thêm ở giữa hai cái này có một cái marker nữa, và khoảng cách giữa cái hai marker này là bao nhiêu? 15 cm, đúng không? Là khoảng cách giữa cái thứ nhất và cái thứ hai. Và khoảng cách giữa cái thứ hai và cái thứ ba là bao nhiêu? 17 cm. Và tôi sẽ nói là bây giờ tôi phát hiện thêm một cái marker nữa, như vậy hệ gen người có 3 marker, và khoảng cách di truyền bộ gen người sẽ là 15 cộng 17 là bằng 32 cm. Điều đấy có nghĩa là gì? Khoảng cách, nếu mà ta gọi mốc này là 0 thì mốc này sẽ là 32. Tuy nhiên nếu nói về tần số trao đổi chéo giữa điểm này và điểm này thì nó vẫn là bao nhiêu ạ? Vẫn là 20, vẫn là 20% xảy ra trao đổi chéo. Nhưng bởi vì tôi phát hiện ra một cái điểm nữa ở đây, và điểm này có khoảng cách ở đây là 15, khoảng cách ở đây là 17, cho nên tôi gọi là bộ gen người có kích thước là 37 cm. Bây giờ về sau các em nếu mà bập bài báo di truyền về các marker phân tử thấy ví dụ bộ gen ruồi giấm, nếu mà quy theo cái SNP là nó có khoảng cách hệ gen của ruồi giấm là 400 cm chẳng hạn, thì ta biết là tổng khoảng cách tần số trao đổi chéo giữa các điểm liên tiếp là khoảng độ 400 cm. Yeah. Ok.
Có nghĩa là khoảng cách giữa điểm 0 và điểm 32 này là 20 hay là 32?
Có nghĩa là ở đây chúng ta không đề cập giữa khoảng cách giữa điểm 0 và điểm 32. Tôi gọi điểm này là mốc là 0, điểm này mốc là 15 và điểm này là mốc là 32. Đúng không? Như vậy là toàn bộ hệ gen này nếu mà dựa vào ba cái marker này thì kích thước về hệ di truyền là 32 cm. Tuy nhiên tần số trao đổi chéo giữa điểm 0 và điểm 32 này vẫn là bao nhiêu? Vẫn là 20%. Cho nên nếu mà tôi cần phải tính cái tần số trao đổi chéo giữa điểm 0 và điểm 32 thì nó vẫn sử dụng con số là 20% thôi. Đúng không? Hiểu điểm này em nhỉ? Okay. Okay. Right. So the same within here, we have the first genetic mark based on the phenotype in tomato. So in tomato we have 24 chromosomes. So in chromosome number 1, we will have the genetic distance between these two pairs is 12 chromosomes, 12 centimogon, right? So the genetic pair is 4 centimogon, 17 centimogon. So that means that the genetic distance of all these markers in chromosome 1 is 12 plus 4 plus 17 plus 16 plus 14. Okay, plus 5, 4, 6, and 14. Okay, this anyone can help me to calculate this. What is the total genetic distance of chromosome 1, please? It's 12 plus 4 plus 17 plus 16.
78.
How much?
78.
78.
Right.
78. So the genetic distance of the chromosome 1 is 78 centimogon. Okay. So that means that the genetic the total genetic distance between two continuous closest traits is 78 centimeters. And this is the genetic map of the human chromosome 12. And the interesting here is in chromosome 12, we will have the same marker between male and female. So this is the genetic distance of the female, and this is the male. We can see that all the markers are the same. But the total genetic map of this in woman is 168 centimorgan, and in man it's just 78 centimorgan. So what does that mean? Can you help me to provide a conclusion? What is this mean?
This means that the centimorgan cannot be used to measure the correct, to measure the precise length and distance, like for example in angstroms or meters or so on between each gene, but rather only the relative.
Okay.
The distance relative to the recombination rate.
Okay. And anything else?
And in women, the crossing over is much more common.
Okay, so that means that in women, normally the chromosome 12 in women is happen, the cross-link is happen more in chromosome 12. The cross-link phenomenon, it happen more in women than men. Okay, and that means that the same chromosome 12, the same marker, the same genetic, the same physical distance, everything, but the chromosome 12 in man is more stable than in woman. In woman, the ratio of cross-link of the chromosome 12 is happen more frequent than in man. Okay. Right. Okay, we come to another very important term is the haplotype. So haplotype is a set of marker between different a set of allele between different marker. So for example, in here we have three marker, which is SNP, single nucleotide polymorphism, SNP1, SNP2, and SNP3. And in SNP1 we have two allele, A and C. SNP2 we have two alleles, T and A, and SNP3 is C and T, for example. So it will provide that if we have in SNP1 we have the allele is C, T, and C, so that means we have a haplotype, which is the genotype in here is CTC for the pre-SNP, and the another haplotype is the for example in here SNP1 is still C, SNP2 is changed to A, and SNP3 is changed to T. So another haplotype. So it's a set of allele that corresponding always linkage disequilibrium to each other. And we come to a new term, high linkage disequilibrium, according to remember to everyone. So for example, in here we have a 1, 2, 3, 4, 5 SNP, which is inherited together as a block. That means there is the ratio of cross-link between those is 0% in here. Okay, so that means each SNP here is a tough SNP. Since we genotype one SNP, we will genotype also each other. And tough SNP is a very modern technique when you want to analyze a genetic profile of an unknown organism. One thing is here, I want to show that about the stroke of the SNP. So normally, the physical distance, the genetic distance is correlate with physical distance. However, it also depends on a source of a SNP. So for example, in here, when we study a species, we have a common ancestor. When we compare between the two SNP with a yellow and green in here, although the physical distance L is really far from here, okay? But we can see that they are in full linkage. So wherever you go, I go. Wherever this one is a yellow, this one is green. And wherever is a white type, this one is also white type. In other words, when we compare between these two, the black one and the yellow one, apparently they are not in full linkage disequilibrium. Okay, so we have combination, we come in here. That because even the physical distance is really close. That because that the order of appear of this SNP in here, the SNP, the mutation yellow was appear in one ancient generation. Meanwhile, the black one have appeared in only this. So that's why it's only here, but not appear in all the other yellow one. Okay. Okay, we come to the next part, which is positional cloning. So what is positional cloning? So the positional cloning is the modern approach where we want to study genes corresponding to a phenotype or disease. So we have two types of approach. First is a functional cloning, which in here we identify the disease first on the function of the disease. So we have to know about this gene, why, what is mechanism behind this gene, what is function of each gene correlated to the disease. And after we know all about the function, we identify about the genes, and last one is the map. However, functional cloning bring also some disadvantage. First, sometimes we have to know we have to know everything about the function of all the gene related to disease in the gene network related disease. And secondly, it's not always precise, since when you identify between the patient and the health control, we see that there's different because they are just two different persons. Sometimes it's not related anything to the phenotype you are doing your research in. So we come to another approach, which is position cloning, in which we identify the disease first on the position of the genes. After we identify the position on the genome, we identify genes and then figure out what is the functional. Okay. So with the functional cloning, that means the protein comes first and we find out about the gene later. But in positional cloning, we identify the gene first and the protein come after. Okay. So how can we launch a positional cloning? So let's hypothesis that we have two individual or two gametes, A and B. We have three markers, M1, three locus, M0, M1, and M2. Each locus have two alleles, A and B. Okay. So after meiosis, we analyze the gamete and we have the ratio of each gamete. We jump type it and have the result, like so in the table. Can you help me to have a conclusion? What is the relationship between M0, M1, and M2 based on this result? Anyone? what is the relationship between these three marker, M0, M1, and M2? So in here, we can see that M1 and M2 are in full linkage, right? Whenever M1 is A, M2 is A. Whenever M1 is B, M2 is B. M0 is not in full linkage with M1 and M2, right? So right now, if we hypothesize that there is a mutation, a gene was causing a disease was happen in this gamete between M1A and M2A and did not appear in here. So this is a wild type version of the allele. So that means that if F1 is A, F2 is A, that means it also carry the mutant, because this marker A in here is linkage with the disease. Okay, so whenever M1 and M2 is A, that means it's a sign for a marker for a mutation disease phenotype here. And if it's the B, it's wild type. So that is how we can perform positional cloning in here. So in fact, nowadays we don't have to perform linkage with 3 or 4 marker anymore. We can perform one single time with 1 million SNP across the genome. So we have a DNA chip that can genotype millions of marker at the same time, millions SNP at the same time. So for example, in here you have observed a genome-wide association study across the genome. So in the vertical axis is linkage between, it actually is a minus logarithm of the p-value, but for the time being, just understand that it's a linkage between that marker and the phenotype. And in the horizontal axis, you can see the position of the SNP across the genome from chromosome 1 to chromosome 22. In here, we didn't analyze the gender chromosomes. So we can see that there is several positions here. Here we have 5 positions that show a high linkage between 2 continuous marker and the phenotype. So that means, and the most remarkable one is this one. So this one is containing in the region a gene that really related to a disease. Okay. All right. So a very quick reminder about the Hartley-Weinberg equilibrium. So the Hartley-Weinberg equilibrium is a principle that was invented by the 2 scientists, Hartley and Weinberg, to calculate the frequency of allele of a population when they are in the equilibrium in genetic genetic term. So, for example, if we have a 2 population follow a Hartley-Weinberg equilibrium, that means that the allele frequencies is not change from generation to generation. Okay, and if we have the allele frequency was not changed, so if we have the marker we have 2 allele, we will have the formula will be p plus q square is equal to p square plus 2pq plus q square, which p square is equal to the individual that have the homozygous of 1 allele, and the q square is the frequency of the homozygous of the other allele. okay, and the heterozygous is 2pq. So, just an example. If we have a population in which the homozygous of a recessive allele is small a is 36%, that means that the frequency of the genotype small a is 36%. And we assume that this population is in equilibrium. So what is the frequency of the allele A in here? Yes, the allele frequency A, Q of A, is equal to square root of the AA, which is 0.6%. What is the frequency of the other allele? So, no. We have P plus Q is equal to 1. In this, we have only two alleles, big A and small a. The small a is 0.6. That means this one is 0.4, right? So P is equal to 0.4%. That means 40%. Sorry. What is the percentage of the frequency of the people have genotype big A and big A? Yeah, so big A is big A because we just calculate the frequency of allele A is 0.4. So like the formula in here, the frequency of the genotype big A is P square, which is 0.4 square is 16%, 0.16. Okay. And the last one is the heterozygous. The heterozygous is 2pq, all right? 2pq is 2 times 0.4 times 0.6 is 48%. So now you can go to ensemble. So in bioinformatics practical, we have identified about a SNP called exercise. So for example, the SNP here, we can observe that if they are in linkage disequilibrium in any study population, type and go. Is 5' UTR on population genetic. And we can see here, for example, this one is have the allele frequency is 0. The T is 53.4% and the C is 40.6%. We can calculate it in here to see that if this SNP is not following the ugly one-back equilibrium. Okay. So there's five conditions that a population must accrue that to be sure that they are follow the ugly one-back equilibrium. First, based on a hypothesis, there is no DNA mutation here to change the allele frequency. because if I change allele frequency, then the stable of the allele frequency in each generation will not maintain. Secondly, the population was quite isolated, so there is no migration. There are no genetic, what we call genetic rift between one population to the other. So I have hypothesis there's no it's quite isolated population. There's no exchanging genetic background with the other population nearby. Three is a random mating. So to be sure that they follow the active web equilibrium, one of the two alleles must be sure that there is no mating based on this allele. Otherwise, one allele will have a bias. It will have more chance to passing this allele to the future generation than the other. Then this condition will be can consider that affect into the genetic proportion of the population. Genetic drift, that due to the small population, some alleles will be eliminated out of the population just randomly by chance. And finally is the natural selection, since one of the two alleles have a more vitality than the others. Okay. Right. Okay, so I will finish this chapter here. We will have a small break, a 50-minute break, until we then we will come to the last chapter related to the epigenetics. Do you have any questions? Okay, thank you very much. So we have a small break, 50-minute break, okay? And then we will come back to the last chapter of today. Thank you. Okay, so let's get back to work. So what is epigenetics? So we both know that the first zygote was formed in a fertilization between a sperm, a male gamete, and an egg, a female gamete. When they combine, they become a zygote, and this zygote will form in the whole organisms. But in organism is composed from many different cell types. So for example, in here, in different species, we will have a different number of cell types. So for example, in the first multi-cell organism, we have around one organism will have around 100 numbers of cells, 100 cells, and there are only two cell types, the inner cell and outer cell. But in sorry, I have a phone call. I'll call you back. So for example, in human, we have around 1 million billion cells. That means around 1,000 trillion cells. And it's belong to 265 cell type. We have a brain cell different with the morphological different with the blood cell. The blood cell with the morphological different with the skin cell, different with the hepatocyte, with the liver cell, different with the stomach cell, and all the other cells. So from one cell, the zygote, we can see that all these 200 all the 265 cell type in our body have the same genetic background because it was shared from the first zygote, right? Only the gamete is a little bit different since the number of genome was decreased by a half, but we will talk about it later. So in here, why all 265 cell type have the same genetic background, but their morphological are so different? So from embryo state, the zygotes here is combination of sperm and egg. We have half of our genome carry from father and half genome come from mother. After a few cell cycle, which is 24 hours per cycle. So in after 5 days, we will have a 32 cell stage. So the 32 cell stage is very small, just a size of tip of a needle. But it has formed into two layers of cell, the outer cell and the inner cell. The inner cells here will develop into all the human embryo, and the outer layers will develop into the placenta. So this is the macro scope of scan of the 32 cell stage. So during the differentiation, this first stem cell will develop into different cell type. So my question is, why all of these kind of muscle, the muscle cells, the nerve cell, cardiac cell, all these types of cells are so different from the biochemical aspect, from the structural aspect. Meanwhile, they have the same genetic background, the same genetic background with the zygote, the stem cell. So when we analyze a different cell type, for example, in here, when we analyze the neuron cell, the cell on the brain, we see that the subset of genes that related to express dopamine was upregulated. That means expressed really strongly. Meanwhile, the set gene related to express hemoglobin and myoglobin was negative. That means they are not expressed at all. Meanwhile, in the blood cell, the hemoglobin was upregulated, and the two other sets was downregulated. And in the muscle cell, the hemoglobin and the dopamine was negative regulated, downregulated, and the myoglobin was upregulated. So the question is how each cell type have some set of genes was turned on, I mean upregulated, and meanwhile the other subset of genes was downregulated. Well, it's due to the epigenetic marker. So to come to the epigenetic marker, first that we have familiar with 5 types of stem cell. First is the totipotent stem cell, is the same stem cell with the zygote. So this is the most powerful stem cell, and they can differentiate into into every type of the cell in an organism. The second most powerful is the pluripotent stem cell, which is can divide into 3 layers: the ectoderm, the endoderm, and the mesoderm, which is the inner cell. And the mesoderm is pretty much like the pluripotent stem cell, can different into different cell type in the human bodies. The third powerful stem cell is the multipotent stem cell, which is can differentiate into a spectrum of cell type. For example, it can be different into the blood multipotent stem cell, which is the bone marrow, can different into white blood cell, platelet, erythrocyte, CD4 cell, and all the factor in the blood vessel. The fourth is the oligopotent stem cell, is just can different into specific range of cells. And the last one is the unipotent. So uni is that mean that it can different into only one kind of cell. Okay, so this is the least powerful stem cell. So when we analyze from different cell type, we can also analyze the epigenetic marker between different cell line. The same phenomenon can happen when we are doing research on the identical twins. So we know that the identical twin is siblings that born from the same zygote, right? One zygote will develop into one individual, but for several reasons, sometimes one zygote can develop into 2 different individuals, And because these two individuals have the same genetic background, so they will have the same gender, same everything. But when we observe the identical twins, we realize that the more time gone by, the longer time gone by, the more differences in the phenotype that we can realize. For example, in here, everyone can, when the twins was in the young age, even their parent cannot recognize the difference between those. But the more they grow up, the more time gone by, there are differences between those, and we can recognize them easily. So research had been analyze the epigenetic marker between the chromosome number 3 in a 3-year-old twin and a 50-year-old twin, and we can see that in the 3-year-old twin there is the epic mark between the twins are more or less the same. So up in the yellow color here, yellow color, that means the gene was not up or down regulate compared between the two twins. But in the 50-year-old twin, we can see that in here, there's some region that upregulate in one person and the others was downregulate compared to the other person. So their genetic background are still the same, but the epigenetic background are more and more different come along with time. And sometimes the differences can show up really significant in the outcome, like this one. So we can see that in this paper, which published in 2005, that although the identical twins is also called as the monozygotic twins because they come from the same zygotes, remember? So in here, there is more and more remarkable difference in the 5-methylcytosine DNA. That means the DNA methylation that we discussed, and histone acetylation, which is histone modification. We'll talk about it later. More and more genetic marker in the year during come along with time. So papers show that that identical twins sometimes have the different appearance, like according to statistical number, 30% of the twins have a different height. And sometimes the difference is so large, like this. So yeah, in case you are wondering, this is a identical twin. That means these two brothers have the genetic background. But you can see very easy to recognize that they are quite different from their height to the body shape to the face expression. So this experiment was performed in the Utah University based on the mouse. Okay, so how can a mouse can determine if the genetic background can affect into the characteristic of a mouse? So if come from a mouse, so in here the two mouse is based on with the same the two mouse in here are raised is the same line, and in terms of the same line, we see that these two small mouse here have the same genetic background. Okay, and they was arise in the almostly the same condition. They are feed with the same food, the same condition of cage, the same temperature, the same everything. Only one difference between the two mouse. In one mouse, they let the mother nurture, that means licking their fur of the mouse. And in the other mouse, the mother was not, the scientist not let the mothers licking the fur of the mouse. So that is the only difference between the two. And they see that when the mouse with a high nurture, that means that the mouse mother usually licking the fur of the mouse here, it seems that the mouse, when they grow up, they seem to be always calm, relaxing, and when their heartbeat is quite steady, and when they heard an expected noise, they are looking forward into the noise, see to check if the noise is the sign of danger. Then if there's a sign of danger, they run away. Very calm. Meanwhile, in the mouse growing up with the low condition of nurture, that means the mother mouse never allowed to licking the fur of the mice. They seem to be always stressed, always anxious. The heartbeat rate is so fast. And when they heard an unexpected noise, they run immediately without checking if there's a sign, is the noise is a sign of dangerous or not. So when they further investigate this situation, they see that there is a gene called the GR gene in here, and it acts like a dopamine to the brain. It's like morphine to the brain. It calms down the brain. And in the high nurture mouse, during a childhood, the GR gene, the more the mother mouse was nurtured its baby mouse, the GR gene was the more relaxing state. The GR gene show up, and it's untwist so large that the transcription factor can come and start to synthesize the GR gene and synthesize the protein. Meanwhile, in the low nurture one, the GR gene seems to close in the very high dense here, and as a consequence, the transcription factor cannot combine and synthesize the GR protein. So the scientists check to synthesize the GR protein artificially and inject it into the stressful mouse. They calm down for around 10 minutes to 15 minutes, and then they become anxious again. And the funny thing is, this GR gene, the whole process that GR gene was untwisted and start to synthesize the GR protein. The nurture condition only working when the mouse was on the early childhood. That means after the mouse had become adult mouse, no matter how oftenly the mother mouse would nurture the babies, the GR gene was not expressed out anymore. So this process only efficiently during early childhood. Okay, so this is example how early childhood trauma can affect into our genome and decide which gene will turn on and which gene will turn off, and also take an effect on our characteristic. Now, we have not performed such experiment in human or not, but based on the same mechanism in all animal, we suspect that in human, maybe the same mechanism will happen. So what is epigenetics? Epigenetic is a study of the regulation of a gene activity and the level of expression that not depend on the genetic DNA sequences. And in gene, we have the term genome refer to all the genes in the nucleus of a species. We have also the term epigenomics to refer to all the epigenetic chains across the genome of the species. So in our program, during to the limitation of the time, we only discussed on three main mechanisms of epigenetics: the DNA methylation, histone modification, and Rna, also called Rna interference. So what is DNA methylation? So combined with histone modification, DNA methylation and histone modification is called the epigenetic marker of the genome. So DNA methylation is a phenomenon called a cytogen. It happens mostly in cytogen, in the nucleotide C of the DNA, that the common number, the position number 5 here was attacked with a methyl group. And this methyl group here was assigned to show that the DNA fragment at that position was close. So for example, in here, in a beginning of gene, so this is the position +1. So this is a promoter region. And this is a start to go into the 5' UTR region of the genes. So if we have a 5' UTR or in the beginning a region of the gene was mostly methylated, it assigns that a gene here was close from transcribe. That means the transcript factor will not binding in here and start to synthesize the genes, start to express the genes. So the DNA methylation was observed most frequent in the region called CpG island. So CpG is the P in here is stand for the phosphate, is referred to the phosphates that linkage between the C and the G. And the CpG island is the reason that CG is appear more frequent than the other reason. So for example, in here, we can see a CG island, which is CG is appear a lot in yellow. And normally CpG island is a sign of DNA methylation within mammals. Statistical numbers show that there's 70 to 80% of the CpG islands are methylated. So how was it occur? The DNA methylation was carried by an enzyme called NDMT1, and it will transfer the methylated to a methyl group to a cytogen's nucleotide to make that C was methylated. And DNA methylation was show up in imprinting, in X chromosome inactivation. So let's take an example like the X inactivation. So we know that in mammals, in human and in mammals, the double X is encoding for is female and the XY is in male. Vice versa, in several type of bird or in chickens, the XX is male and XY is female. So in the XX individual, the X inactivation is that in the two double X, we have only one X was activated. The other X was inactivated due to amount of DNA was methylated too high in X chromosomes. So, for example, in the case of the calico cat, okay, so we know we study this in high school. We know that the calico cat is a female cat, and because of the fur, the gene was encoding for the color of the fur is located in the gender chromosomes, right? But why in the calico cat we have a fragment is yellow and the other fragment is black? Meanwhile, the genetic background in the yellow and the black are the same. So the answer is because of the X inactivation process. So in here we have the 2X, one is from father, is one from mother. And during the zygote development, one chromosome X will be activated and the other will be inactivated. And the choosing which one is activated, which one is inactivated, was total randomly based on the cell. And as a result, part of the chromosome, part of the cell where the chromosome carry the black color pigment is inactivated will have the yellow color, and the group of cell when the X carry the yellow color was inactivated will have the black color. How about the white? Is that both X was inactivation? No, actually the white color is come from epitasis that we have discussed in chapter 5. So this is pair is encoding for another pair of allele located in the different chromosome that prevent the both X express the color of the gene. So in this one, it's have a white color. So let's compare the DNA methylation profile between the pluripotent cell and the unipotent cell. Pluripotent cell, as we discussed, is much more powerful than the unipotent cell, which can differ only to one type of cell. So we are looking at the CPG island of the gene. So we can see that in here we have beginning of the gene. So we have the opening the start codon here, ATGs. So this is polypeptide started. So this red here is the 5' UTR. And this region here is promoter. So this is the upstream region of the gene. You can see a lot of CG island in here. But when we compare DNA methylation between the pluripotent cell and the unipotent cell, we can see that in the meanwhile in the pluripotent cell, most of the CG was unmethylated. Meanwhile, in the unipotent cell, most of the CG here is methylated. So that means that this gene in the unipotent cell is turned off compared to the pluripotent cell. So the methylation and research in methylation is play a critical role in cancer research. So let's see how DNA methylation can play a role in contributing in cancer cells.
In the cell, there are many methods for controlling how DNA is read and expressed. Several of these methods, called epigenetic modifications, can turn specific genes on or off without altering the DNA sequence. One of these methods is DNA methylation, which occurs when a methyl group is added to a DNA base. When this happens, additional proteins can no longer bind to this region of DNA. This prevents the DNA from being transcribed into RNA. The gene is turned off. Normal methylation patterns are important for the development and function of healthy cells. In cancer cells, abnormal DNA methylation patterns are seen. Genes that would normally suppress tumor growth see higher levels of methylation and are turned off. At the same time, genes that increase tumor growth, called oncogenes, see a decrease in methylation and are turned on. This results in unregulated tumor growth.
So the next epigenetic mark is the histone modification. So we have studied in chapter 1, we know that in eukaryotic cell, the chromosome was composed by a fragment of DNA was wrapping around a protein called histone. And an octamer of histone was composed by 2 subunits of H2A, 2 subunits of H2B, 2 subunits of H3, and 2 subunits of H4. Okay. And in the octagon here, we have each subunit will have a tail. And if we have a tail here, depend in the tail was a modification as acetylation, while acetyl group will attack into the tail. Methylation, while methyl group was attack into the tail, or ubiquitination or phosphorylation. The group of the histone will carry a different property. So, for example, in here, if we have a H3 histone mark called H3K27 AC, that means in the subunit of H3, in the amino acid 27K stand for lysine, was acylated. So, histone modification. So we have a histone here, like octamer, right? So in here we have several one, another one, because octamer is actually 8 subunit. Okay. Okay. So we have DNA come and wrapping around in here. And each subunit will have a tail. So for example, here, if we have in here acetylated, So the acetylate group will take their electron out of the histone. Okay. So yeah, it will put the electron out of the acetyl group and put it into the histone group. Okay. So as a result, when we have an acetyl group here, it will make the octamer more negative charge. And since the DNA was which charge? The DNA was negative charge in the neutral pH. Remember? So because if negative charge, so and the octamer was more negative charge, it will lose the attack binding into the acetyl group, and as a result, it will create more space to the transcription factor. This is transcription factor. Come and binding into the genes have to upregulate expression level of the gene. Meanwhile, for example, in here I have a different octamer. So I only draw for the and we have the DNAs common wrap around here. So now, for example, we have a histone modification is a methylation group. Methylate. And normally the methyl group will suck the electron out of the octamer. And as it sucks the electron out, it will make the octamer more positive charge. Right? So since it's a positive charge and the DNA was still negative charge, the iron bond will make the DNA strand wraps more tightly around histone. So it leads to the transcription factor cannot bind into the gene region. So it comes to the down-regulated. Expression. Okay.
Actually, so are there methylated histones?
I'm sorry, what is the question, please?
So how exactly the histone gets modified so as to wrap the DNA more tightly?
You want to ask about the mechanism the histone will modify, or you want to asking about the mechanism of the DNA will wrapping around?
How the histone is modified.
Okay, so we are talking into it. Okay, so how the histone was modified? So it was modified based on different enzymes. For example, in here we have different enzyme like HAT, which will make the acetylation for the histone protein. And one we have different enzyme called HDAC, called deacetylation, to remove the acetyl group out of the histone protein. And each of histone modification, we have different enzymes to come with the process. For example, for methylation, we have HMT for methylation and HDM to demethylation for the protein. And depend on the different type of, sorry, I will come back. Okay, so in each type of modification, we will have different enzyme will come to upregulate. That means that more methylate, for example, for methylation. Sorry. Okay, I will. Okay, so for example, in here, when we have a fragment of genes here, so in here we have a DNA fragment that wrapping around the octamer, the histone protein. So if the cell decide that this gene need to be down-regulated, so the complex with the HDAC will come and H more deacetyl more group in the histone tail in here, and because the more histone tail would deacetylated, so the more, like we can see in here, the more histone tail was deacetylated, so the more negative charge the histone protein will lose, and as a consequence, the DNA will wrap higher in the octamer, and the transcription factor binding cannot come and start to express the gene. Meanwhile, if a gene was a cell will decide a fragment of gene need to be activated, so a subset of enzyme HAT will come to adding more acetyl group in the histone tail to make the histone more negative charge. So the DNA will lose more tightly, more relaxing state in here. So it create space so the transcription factor can come and bind to the DNA, and that's how is the HDAC and the HAT can have the cell to activate and represent the gene expression level of the genes.
Epigenetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
So the last mechanism that we discussed today is the iRNA. So if the DNA methylation and the histone modification was interfere on the transcription level of the gene in terms of the gene before the transcription, Rnai or Rna interference interfere in the expression level of genes after the transcription had been of the gene was happening. So the process was happen in the process here in a normal condition when a gene would decide to express out. The mRNA, the transcription will happen inside of the nucleus, okay, and the mRNA synthesized going through a major process to remove the intron at the 5' cap and pull A tail, getting out of the nucleus into cytoplasm and attack into ribosome to start the translation. However, the iRNA will interact after the mRNA will be synthesized. So the RNA will combine with the mRNA here, and if it have the sequence fit with the mRNA, it will use a special mechanism called DICE, RISC, to cleavage the mRNA as its form, and because no mRNA, so no translation will be processed, so no protein product will be synthesized. Okay.
RNA interference was popularized by work in C. elegans. When long double-stranded RNAs were injected into a worm's gonad, a standard way of introducing transgenes into worms, they blocked the expression of endogenous genes in a sequence-specific manner. In eukaryotes, most protein-coding genes are transcribed by RNA polymerase II, which generates pre-mRNAs that are then processed to form mature mRNAs. These mRNAs are then transported from the nucleus to the cytoplasm, where they are translated. RNAi can regulate endogenous gene expression. RNAi can be set in motion by genomically encoded short regulatory RNAs known as microRNAs. In algae, worms, and flies, RNAi can be activated by endogenous transposition. In plants and cultured insect cells, RNAi also has a role in antiviral defense, in which viral double-stranded RNAs are targeted for destruction by the RNAi machinery. When long double-stranded RNAs enter a cell, they are recognized and cleaved by dicer, which is a member of the RNase 3 family of double-stranded RNA-specific endonucleases. Cleavage by dicer creates short double-stranded RNAs that are characterized by 2 nucleotide-long 3' overhangs. These are called small interfering, or siRNAs. siRNAs can form a ribonucleoprotein complex called RISC, or RNAi silencing complex. This complex includes slicer, an argonaute protein with an RNase H-like domain called PW. RISC first mediates the unwinding of the siRNA duplex. A single-stranded siRNA that is coupled to RISC then binds to a target mRNA in a sequence-specific manner. The binding mediates target mRNA cleavage by slicer. The site of the cleavage falls in the middle of the region of siRNA complementarity. The cleaved mRNA can be recognized by the cell as being aberrant and then destroyed. This prevents translation from occurring, silencing the expression of the gene from which the mRNA was transcribed. In plants, the aberrant RNA that results from the RISC-mediated cleavage can also serve as a template for RNA-dependent RNA polymerase, or RDRP. This process relies on unprimed RNA synthesis, in which the aberrant RNA is used as a template. The resulting double-stranded RNA is a substrate for dicer activity, which generates more siRNAs. In some organisms with endogenous RNAi mechanisms, for example, fungi, plants, worms, and mammals, RNAi also involves another amplification step. In this step, single-stranded siRNAs not associated with RISC bind to their target mRNAs in a sequence-specific way and serve as a primer for RDRP to polymerize the antisense RNA strand. Such specificity is intrinsically sensitive to natural sequence variation. The double-stranded RNA molecule that is created serves as a substrate for dicer, which cuts it into siRNAs. In turn, these can either unwind and prime RNA-dependent RNA polymerization, or together with RISC, mediate the cleavage of target mRNAs. This amplification, coupled with RNAi spreading between cells, is thought to underline germline transmission of RNAi in worms. RNAi spreading has also been described in plants, but not in mammals.
Okay, so that is the end of today's section. So now I will have a quick recap on what we have studying from the beginning and what we should remember. All right. So in chapter 1, please remember for me the structure of DNA. So DNA was composed by is DNA a polymer of a monomer called nucleotide. And nucleotide were composed by 3 main parts: deoxyribose, that means 5 carbon sugars, a phosphate group, and a base nitrate. And please remember what is the 5' and what is the 3' direction of the genes. And we have to remember the 5' base and the structure of 5' base, which one is a pyrimidine and the protein is, and what is the content of the chaga group. A is always linked with C, and G is always linked with C by 2 hydrogen bond. Okay, so we will have also to remember the differences between DNA and RNA. So DNA was composed by deoxyribose nucleotide, and RNA was composed by ribose nucleotide. And DNA was the inherited material of most, like bacteria, cell, virus. Meanwhile, RNA is just a genetic material for some type virus. So the DNA is more stable. Meanwhile, RNA was less stable due to the hydroxyl group is more susceptible to hydrolysis. And while DNA was mostly double strand, RNA is mostly single strand. Okay, and the length of DNA normally is longer than an RNA. Okay, we will have to remember the what is difference in the structure of the virus genomes, the prokaryotic genomes, and the eukaryotic genomes. So in virus, its genome can be DNA or RNA, can be a single strand or double strand, can be linear or circular. Meanwhile, in the prokaryotic cell, mostly the DNA is double strand DNA, of course, but mostly circular. Okay, and in the interact with protein to form a complex of protein and DNA. And in the eukaryotic cell, DNA is the linear, different with prokaryotic cell, it's mostly circular, linear and associated with the histone protein. Okay, we have one octamer nucleus core were composed by 2 subunit H2A, 2 subunit H2B, H2B, 2 subunit of H3, and 2 subunit of H4. And the histone protein H1 is the linker called the linker histone. Okay, and we have to remember about the endosymbiotic theory that in the eukaryotic cell, in plant and in animal cell, beside of the nucleus genomes, we have also the mitochondrial genomes, and in case of the plant cell, we have also the chloroplast genomes. In this organelle here have its own genomes, and the main differences between the human nuclear genomes and the mitochondrial genomes. Okay, so the main difference was here. I will not repeat it. You read it. So in next chapter, what we should remember is on DNA replication. Okay, so in DNA replication, it follows the semi-conservative rule, which means that one strand of the parent DNA strand was used as a template to synthesize the new DNA one, and the different enzymes that participate in the DNA replication, which is the helicase, binding protein to make the separate both DNA strand was not attached to each other again. Primase, which are the RNA primer in the template. DNA polymerase, which is start to synthesize the newly DNA strand from the position of the RNA. Then exonuclease will remove the RNA primer and replace it by the DNA, DNTP. And finally, the ligase will join the discontinuous DNA fragment together. And we have to remember about mitosis and meiosis. What is the differences in the mitosis and the meiosis, what is the cell cycle checkpoint, and how many cell cycle checkpoint we have in the cell cycle. Okay, and the comparison between mitosis and meiosis on the chapter 2. So chapter 3, what we have studied. Chapter 3 is about transcription and translation, which we have discussed with each other, how transcription and translation happen differently in prokaryotic cell and in eukaryotic cell. So how would the gene was organized differently in the prokaryotic cell and the eukaryotic cell? What is template strand? What is coding strand? So we will repeat it in the exercise tutorial class. What is the difference between DNA replication and RNA transcription? So in DNA replication, remember that both strands in the double strand is a template. Meanwhile, in transcription, only the 5' to 3' DNA strand was used as template, because RNA is like DNA polymerase is always working from 5' to 3'. So in here we have the primer, the RNA primer was required in DNA replication, but not required in transcription also. And the base pair, the ingredient we use is ATGC in order to make a DNA strand, and in transcription in order to make RNAs is AUGNC. Okay. And the gene structure in prokaryotic cell is most of the genome of prokaryotic cell was encoding for protein, and different gene was have the similar function, was organized as an operand, and they share the same promoter. Meanwhile, in the eukaryotic cell, one gene have only one promoter, and in the structure of gene where they have also intron, which will be removed out of the structure of mRNA during mature process. So we have in eukaryotic cell splicing process, which is remove the intron out, and we have different way of splicing, leading to different variation of the mRNA for the same one genes. Okay. So translation, you don't have to remember the genetic code, since I will insert this into the final exam for you, so you don't have to remember it. Okay. So that's it. In chapter 4, what we have discussed. So in chapter 4, we have discussed with each other on the genetic mutation. So we have 2 types of genetic mutation. We categorize it to a level in up to chromosome level. We have the chromosome structure mutation, which is deletion, duplication, inversion, insertion, and translocation. We have chromosomal number mutation, which is aneuploidy, which is adding or deletion of one or several chromosomes, and polyploidy, where most happen in plant, while extra set of chromosome was added. And down to DNA level, we have the genetic mutation, where we have also insertion, deletion, duplication, inversion, and substitution. Okay. And in genetic variation, please remember what is the VNTR, what is the SSR, what is the DNA microsatellite marker, and what is SNP. Okay, so you don't have to remember how to calculate the PLP, the probability of paternity test. So this part is just provide further information for the student who interested in how the paternity test was carried on. So in chapter 5, we have to know about the Mendel law, 3 law of Mendel. So three law of Mendel is, where's the slide? So we have a law of domination, we have a law of segregation, and we have the law of independent assortment. And we have also several exceptions of the Mendel rule. We have mitochondrial DNA, which is the gene was located in the mitochondrial genomes and is inherited mostly maternal. We have incomplete dominance, where dominant alleles were not totally dominant against the recessive allele. We have co-dominance, where we have one allele is recessive, but several alleles is both dominant. We have multi-allele polygenic trait, where gene was encoding for trait was encoding by several pair of locus with epitasis, while a pair of locus was dominant against another locus. Okay. So today's section we discuss with each other on linkage disequilibrium. So in linkage disequilibrium, remember what is the main difference between the proportion of crosslink and the genetic distance. Remember that a proportion of crosslink cannot overpass 50%, but meanwhile, the genetic distance can depend on the number of chromosome set, like in here. Okay, and we have also studied on what is the position cloning. So in short term, is in a nutshell, is a method of research which we find out the function of the gene after we know about the position of the genomes that corresponding for one phenotype. And we study about the RT1 back equilibrium, which is mostly like the knowledge in high school, which refer to the population genetic. When a population is in equilibrium, that means they will have the homozygous of the two will be the square of the allele, and the heterozygous will be two times the frequency of each allele. Okay, and in last one, in the epigenetic chapter, we just remember that different cell line in the organism will have the same genetic background, but different gene set will be choose to express based on the epigenetic marker. So we have three main mechanisms of epigenetic marker. We have DNA methylation, where the cytosine in the DNA will methylated, and normally if a fragment of genes was highly DNA methylation, highly DNA methylated, so the gene will be downregulated. So we have also histone modification, which the histone will be adding to group to upregulate or downregulate the gene expression level of the genes. And the final mechanism is Rnai, which is the mechanism where the Rnai will combine with the messenger mature mRNA, and the risk will destroy the mRNA since it will match with the sequence of the RNA, and no mRNA, that means no protein. Okay. So do you have any question related to today's chapter, as well as any chapter in the genetic subject? Any question? No one have question? Okay. Yeah. Okay, so if you have no question, I have a question for you. So every year, I will always ask the student comment on the quality of the lecture and on how the knowledge was transferred, whether it's the knowledge was transferred too quick. That means I say too quick through several parts, or too slow. That means that I understand that there's several parts in here that you have been studying in high school. So do you have any comment, any suggestion for the improvement that I can make, so for the better quality of the slide for next generation of students? Because I have already made a lot of modification in the slide. You can compare with the slide you have model, but depend on each generation, they require different part. So so the slide I use here is not the slide I using previously. I don't see Bio2 on my Moodle. So on Moodles, you can access into Moodle here. So you can log in your Moodle account, and the course Bio 1002 genetics was here. And if you have a problem into access into the course on Moodle, please contact with the GED, the General Education Department, to see that if the secretary here there can help you to access into your Moodle account. Okay. Any suggestion? Any comment? So sometimes I heard that maybe some student can say that a lot of the part of knowledge here was have been already they have been already studied in high school, right? So maybe I should overcome those part and adding new part. The summary slide. Actually, I don't have the summary slide. The summary slide is just a quick recap on the slide I have presented to you. So it's located somewhere in chapter 1, a few slides, in chapter 2, a few slides. So you can look at in each chapter to see what is the slide that most important. So in here, I want to say that maybe there's some students think that there's several parts that have been taught in high school, but because you know that in here you are a class with more than 500 students. Yeah, so the knowledge background is quite variable between you. So the idea of the organizing the class is to make everyone moving the same with each other, to push the whole class moving together, not like a group of students who expertise in biology is moving really fast and the rest was keeping behind, very long distance behind. So it's not a way to organize the class, because you are too many and the level of you is quite variable. So so it's had to be organized like that in order to make all of you moving together. So it's the idea. Okay. Can we have a more detailed survival kit? I think the survival kit in here is detailed quite enough. Survival kit. Well, I think the survival kit here is detailed enough. So in order to have a minimum result to pass my course, So first, attend to all the course. Don't miss any point out just because your absent is any section, like in tutorial or in the lecture session. Remember to submit your bioinformatics practical course, and you will have to study each of the part was listened here in each chapter, and remember the exercise. That's all. Okay. So, yeah. Okay. So, any other suggestion? Any? What is the detail of? Suggest a mind map to recap. Well, I think it's a good idea. Maybe I will deploy a mind map in the next year for later generation. Thank you. I think this suggestion is quite useful. Any other suggestion or comment? Okay, so if you have no more question and no more comment, I want to thank you very much for your patience. You have come along with me in the 10 session, and two last session is really long in I don't know, 3.5 hours, nearly 4 hours, and you are still with me to until the end. I still see 212 students here, so wow. Okay, thank you very much, and good luck for your final exams, and I will see you all in the exercise tutorial class. Okay, thank you very much, and see you later.
Teacher, I have a question.
Yeah.
I remember that you had mentioned us about the tutor bio, and in that class we will have exercises. So
yeah, exactly.
Will we have a tutor for it, and about the exercises?
Yeah, the exercise will be handled. Wait me a minute. I will open the. Okay, so exercise. Okay, so in the tutorial class, which will be organized in the middle of March. In more 2 weeks more, I think will be the first tutorial class. So we will have 5 tutorial sessions, and in each session we will handle 1 chapter. So we have 5 chapters here. Okay. So first of all, in the tutorial class, there will be the attendance check. No matter what the session was organized online or offline, there will be an attendance check for each chapter. So first, present you there. Do not take any penalty because of your absence. And secondly, if you will be able to handle any exercise in here, please volunteer to handle the exercise, and you will have your reward point. In case you cannot handle any exercise, wait for the other to handle exercise and put it and try to understand the solution. If you don't understand solution, please ask the supervisor at the time. So there will be a different lecturer participate in the exercise tutorial class. So I will corresponding for 3 class, and there will be 4 other lecturers participate in. So if you have any difficulty in handling any exercise, please ask your support your lecturer at that tutorial class. Okay. Any question? Am I answer well your question, or you still want to ask about something else?
Yes, I satisfied your answer. Thank you very much, teacher.
Okay. Okay, thank you very much. And see you all in the tutorial class. Thank you.
Thank you, teacher.
Thank you. See you.
Yes, sir. See you again.
Thank you, professor.
Thank you. Thank you, teacher. Goodbye.